we've never talked about before, because it's a little bit challenging. And so we figured that this was a safe space for us to talk about it, because we had something amazing happen. And so we'll start with Dr. Rahal introducing Dr. Porter to tell you a little bit about it.
All right, everyone. So I know Dr. Porter just introduced himself, but for those of you who are just coming in, I will do another introduction. He's my mentor. He's taught me everything I know about SLOS. So far, I'm learning a lot from you guys as well, but Dr. Porter is a senior investigator in the Intramural Research Program at NICHD. He's also the director of the NICHD Molecular Genetics Core and an associate scientific director for translational science. He was the clinical director of NICHD from 2010 until 2022, and his group is involved in multiple disorders. One of them is Smith-Lemli-Opitz syndrome and has been for years, and other disorders such as Niemann-Pick disease type C and other disorders. So here's Dr. Porter.
So, you know, last night I'm putting together the slides. Gretchen wanted them a lot earlier. And I was trying to think about, you know, what the title is, but the title actually relates to that committee that was sitting up here.
Stand by the podium.
And I stand by the podium. Yep. Okay. So how do we answer those questions that you have? And it's based on experience. It's based on clinical trials. It's based on the efforts that the kids and the parents and the families made to come into those studies. That's how we get the answers. That's how we continue to get answers. So I didn't come up with this, but the concept of pay it forward. Okay. How can we as a community help those kids and help those families? Also help you, but help the future kids with SLOS. Okay. What we learn may help your child. That's the goal of everyone in this room. Okay. But what we learn will help other children in the future. I think Marty mentioned, how much did you have when Chandler was born? 0. We have a lot more now. And it's because of those families that we can help you. And what we want to do is help other children going forward. Okay, participate in research. There's all kinds of levels. There's not one right fit for everyone. You all have different life circumstances. Registry surveys, this is something we were talking about the other night. They can range from simple to complex, but they give us information. The natural history study got referred to that we've been maintaining at the NIH since 1998. It includes a biomaterial collection. That's not necessarily easy when I start talking about cerebral spinal fluid. I'll come back to that in a little bit. Much easier to talk about getting a blood draw. Clinical trials. They are essential in my mind. Okay, we want to know a drug actually works and has an impact. Okay, why do we need participation in clinical trials, natural history, therapeutic trials, etc.? We can get a lot of information. I mentioned earlier, I have a laboratory. I have a number of laboratories. We have mice with Smith-Lemli-Opitz syndrome. They're okay. They can answer some questions. They can't answer all of them. I have a picture of cells here. These are iPS cells, human iPS cells that we can turn into neurons. We can answer additional questions with them. But we can't answer all the questions. How do we translate that information to the patients? And that's the concept of moving preclinical to clinical or translational medicine. It won't work if we only have basic science. It won't work if we only have clinical science. It only works when we put the two together. Okay, so biomaterial. Let me start this off going easy to hard. okay? Sort of a harsh term, biomaterial, but that's samples, put whatever name on it, but to scientists, it's biomaterial. We get it in our natural history trial. The most value comes from well-phenotyped individuals. What do I mean by well-phenotyped? And we sort of were talking about the variation of the phenotype. What does your child have? What are the problems that they have? What are the malformations that they have? What is their biochemistry with respect to their sterols? What are their exact mutations? How can we start to put these things together? Blood and urine, that's pretty simple. Everybody gets blood tests. Urine, well, that's pretty simple, usually. Can be sometimes challenging. It's actually challenging, fairly challenging in some kids to get a good sample. Cell lines, skin fibroblasts. We do a skin biopsy, okay, when we're seen. But if your child's going into surgery, with planning, we can often get the surgeon to send us a small piece of skin. It can be shipped through the mail, and we can, with your consent, we can grow up a cell line. It serves as a resource for future studies. That's how we get those induced pluripotent stem cells to make neurons. But technology is changing. It's not only limited to neurons. You could make blood cells, you could make liver cells, you could make different kinds of neurons. They're a resource for the scientific community. Cerebral spinal fluid, believe me, I spent a lot of time talking about this, okay? Nobody believes me that in a young, skinny child, it's easier than a blood draw. They're not doing this on you, okay? Why? What was that? Well, I do a lot sedated too. And we'll do blood draws sedated if we're sedating. But I do a lot of LPs on kids that are awake if they are at a developmental level where we can do it. But it's scary to parents. It's not part of everyday life. But why? You know, we have problems in multiple organ systems, liver, et cetera. But what's going on in the brain? Okay. What's going on in the nervous system? That's separate from the rest of the body. The closest you'll let me get is cerebrospinal fluid. And I'll show some examples. We can find biomarkers. We can start to understand stuff from the cerebrospinal fluid. It's not for everybody. I understand that. But to those that are willing to let us do it, and we know the frequency of adverse events, believe me, it's great to do it in kids because it's really low frequency of adverse events. We've literally done thousands because there's another disease we deliver the medicine by an LP. We've literally done thousands and can tell you what are the risks, are actual risks of an LP. Here's some examples. So biomarkers in CSF. I pulled from another disease I studied. This is the one I referred to earlier. It's a neurodegenerative disease. The neurons actually die and the kids degenerate over time. The marker doesn't matter. We have multiple ones. This is just a marker of neuronal damage. I don't think I have a slide here. I mean, a pointer here. But in that top graph, you can see non-NPC versus NPC. This marker is markedly elevated in the cerebrospinal fluid of those kids, okay? That tells me that there's neuronal damage going on. I sort of knew that already, but now I have a marker that I can quantify. There is a drug that can be used. So I split it out. It's miglustat, just abbreviated here. No miglustat versus miglustat. And you can see that the individuals that are on miglustat have a lower level of this marker of neuronal damage. Suggests it's still occurring, but it may be occurring at a lower rate. Okay, so that's a biomarker. How do you connect that with an actual improvement in the patient? Just real quick, in the lower, there's some of our work and some work by Mark Patterson on that same drug. We were able to show that it delayed swallowing dysfunction in these kids. He was actually able to show it increased lifespan in these kids. Okay. Problem is that took over a decade to get that data and to see it. this disease progresses over years. The biomarker doesn't take decades necessarily to change. So it gives a clue that you might be on the right track, that you might have a drug that will have a positive clinical effect. We haven't done as much work with SLOS, but this is an example of a biomarker in SLOS. It's a protein called GFAP, has to do with the astrocytes, another cell type in the brain, seem to be putting out more GFAP, indicating that there's something going on in SLOS that activates that cell type. It's increased in SLOS compared to a sample of children that don't have SLOS. You cannot collect cerebrospinal fluid in a normal child. So this just happened to be individuals who have autism spectrum disorder, but the samples are collected the same way. And that's important when you start to look at biomarkers. We actually went back to samples that we had in our freezer from our simvastatin trial that we did probably a decade ago now. And what you can see here, I guess I shouldn't do that. The levels are actually lower in the kids when they were on simvastatin than in the placebo, suggesting that we may be seeing a marker that might respond to a therapeutic intervention. That in my mind was a proof of concept trial. We had a suggestion, maybe of statistical improvement. I don't think we had a clinical improvement. So I don't tell people to go on simvastatin. It's a proof of principle that the concept might work. It's just the simvastatin, you can't push the dose high enough, I think, to make it work in the kids for a clinical effect. Okay, natural history, relatively easy. Registry, really easy. CSF, okay, I'm talking to you for a while about this, and a number of the parents in the room have gone through that talk with me. This one's a harder one. Legacy donation. What do I mean? After your child dies, donating their tissues to support research. Acute death in SLOS, I mentioned it earlier. We all know about it. Okay, and I know it's been on the internet and a number of families that have raised it lately. Why? I don't know. I know in the few cases where we caught what might have been going on, it may be a lung issue. That's what I've seen in a couple of cases. Maybe a metabolic issue. That's what we maybe saw in another case. But we don't know why. This is where a legacy donation could help. If we had good, and we went to the death records. Samar talked about this yesterday. Okay, maybe the death certificates would help us understand. They tell us they had Smith-Lemli-Opitz syndrome. They didn't help. This is not for everybody, but if it is something that you would want to do, this is something that can be done that maybe will allow us to answer this question, why? And if we know why, we might know how to try to prevent it, or we might know a better way of dealing with it than the frantic call from the emergency room or the PICU. Okay, why else? Research, okay? I talked about translation from those preclinical models. I can take apart a mouse, I can study the brain of a mouse. Or in cell culture, I can look at human neurons, but is what I'm observing there what's happening in a patient? How do I translate that knowledge to say, yes, this is something we really need to understand because it is happening in the patients. So that will help. Technology has exploded. Okay, it continues to explode. Put a picture down here. It looks like artwork. This is called a Tisney plot, okay? Each one of those color clusters are a different cell type in the brain. This is a picture of cell types of the brain. So each, there's astrocytes, there's oligodendrocytes, there's different types of neurons, and all of them are represented by a different color. Each one of those dots is a single cell in the brain. This happened to be a brain of an Alzheimer's patient. I can go into that single dot and I can see what genes are being expressed in that cell. And I can compare it to an individual. In this case, they were comparing to the individual who didn't have Alzheimer's disease. This individual had Alzheimer's disease. That technology looks really wild, but it's gotten commonplace. Their technology is moving beyond that. So those, if we have good pathological, using a medical term, pathological material, but we have good samples, these are questions we can start to ask in Smith-Lemli-Opitz syndrome. Ask, what is actually going wrong? How do we answer that question between function and neurodevelopment issues? What might be fixable? So it's an increased understanding of pathological processes and mechanisms. And as a lab rat, I'm going, if I understand that, can I figure out how to treat it or correct it? Okay, legacy donation. It's not easy to think about. These conversations are not easy, okay? They're not easy for us physicians. They're definitely not easy for the families and the parents, okay? Or the individuals themselves, if they're at a level of functioning where they can think about it. It is not for everybody, okay? You should not feel guilty if you say, this is just not for me. This is an individual family decision. It is a gift to the SLOS community and future children. But it only works with planning. We get that call in the middle of the night and said, do you want this tissue? And we're going, huh? I mean, it's not collected right. We're just sitting here, okay, sending something across state lines tends to be problematic when it involves body parts. It doesn't work. And I think that's what the next talk is going to be. If it is for you, if it is something that you can consider as a family, it needs to be planned. It can't happen in the acute circumstance. It's too emotional at that point in time to make those decisions, to make them with everybody agreeing. Mothers and fathers got to agree on this, okay? It needs to be planned, and that's where I think the next talk comes into. If this is something for you, it's a benefit to the community, but it needs to be planned. I don't know if anybody wanted questions or where are we going, right? Yeah.
State Chandler died before I did, because otherwise I'd have to have my son donate her tissue. But if someone dies in Texas, who gets the tissue? How is it sent? Or you just have to sign up
Hold
first?
that question. So there are experts that deal with this.
Oh, they're going to talk about it more.
So,
okay.
Yeah. This is one of the importance. I got the lead off position to tee it up.
Yeah,
but
yeah.
Now, how do you make it work so that I, as a researcher, can then get that tissue type of thing? Okay.
Well, it reminds me of cremation. I'm signing up for that, and then you got to pay for it. You got to sign up with a certain group and make sure when the time comes, everything follows.
Everything falls. Believe me, I think we've all been in the situation where we get a call and it just doesn't work. We can't tee it up at the time. We can't bring the right resources to bear. Okay.
This might be, if not all the way, that's just fine. But
This goes back to the placenta in Texas. So I gave my placenta for my second child.
Okay. I was like, placenta in Texas, sorry.
And I don't feel like it's been used. And I know that like we were at OHSU and that's a different program and all of the pieces there. Because NIH is national, is there, if this process goes through, will it all be connected to a live? I mean, I know there's always chance of things and funding, but is it the way that you're going to show us how to do it, it will all go to an active program?
There are repositories, and Maryland Brain and Tissue Bank is one of them. They know how to do this. And I, as a researcher, I've done it. I can't do it with SLOS because I don't recall being any samples there. But with the other diseases I've studied, I can go into their database. I get permission. I mean, they filter it, so it has to be legitimate researchers. I can go in their database and see what they have. Okay. And that's huge because that's not only me who might know who's in, we're not supposed to, but it's a small community, might know that there's a sample available, but other researchers that are just coming into the field and say, oh, I have this interesting question, maybe I could answer it. Okay. Yeah. So
it's not
these
an NIH repository, it's used more so.
Yeah, it used to be supported by the NICHD, but I don't think NICHD they removed it from the name. But these are central repositories. A lot of diseases have it. This is one that I've worked with in the past. That, but they do make things available to the research community, broad research community.
My name is Tom Blanchard, and I am the director of the University of Maryland Brain and Tissue Bank. One of my staff members, Grace Flannery, was originally going to present because I did not think I would be available. But again, my name is Tom Blanchard. I'd like to thank you for the opportunity to speak to your group. This is important for us to get the word out about what we do. And I realize this is a subject that's a little uncomfortable and not many like to think about. But it's a potential fantastic resource for researchers and those that come after us. I'd like to talk to you today specifically about why our brain bank is uniquely situated to help the SLO community and those with other rare diseases. Next slide, please. So there are many brain banks, actually. Most of them kind of fly under the radar. The NIH funds what they call the Neurobiobank, and it was established in 2013, and its goal was to recruit and collect I'm sorry, it looks like I'm getting word that you cannot see my slides. Okay, thank you. So the Neurobiobank was established with the purpose of getting human neurologic tissue out into the research community to facilitate research. Next slide, please. So the Neurobiobank is actually a network of brains that had existed for many decades. Our bank has existed since 1991. And it's one of 6 banks that collectively makes up the Neurobiobank. And then, as I said, in 2013, they kind of brought us under one umbrella with one unique mission. We still retain some autonomy, but we work together as best as possible to unify our methods and protocols. Next slide, please. So the banks within the Neurobiobank have this mission to collect, store, and distribute brain and nervous tissue from all manner of neurologic disorders. And most of them really do focus on adult diseases, dementia, epilepsy, a lot of psychiatric disorders. But the University of Maryland is fairly unique. And as was said in the previous presentation, the University of Maryland was funded for many years by the NICHD. Next slide, please. We were founded in 1991 with funding exclusively from NICHD. It was dedicated to collecting, storing, and distributing tissue for not just neurologic disorders, but movement, developmental, metabolic, behavioral, and psychiatric disorders in infants and children. And although we became part of the NIH Neurobiobank in 2014, our mission has expanded, but our previous mission has not ended. And so, yes, we do collect adult onset neurologic disorder tissues. We continue to receive a good amount of our funding from NICHD, which allows us to continue our interest in collecting either those diseases that occur uniquely in childhood or those that have onset in childhood. Next slide, please. We collect tissues from all over the United States. We are in Baltimore, and as such, the bulk of our tissues tend to come from the Northeast and from the Great Lakes region. But we can collect tissues from any state in the Union. And this is simply a diagram from 2019 showing you the number of families that have pre-registered with our bank. And so when the time comes, they would alert us, and then we would put things into action. Next slide, please. So there are many benefits to donating tissue to the University of Maryland Brain and Tissue Bank. Again, this is important to the NIH to get these tissues collected and distributed. And therefore, the NIH covers the cost. There is no cost to a researcher to obtain tissue from any of the banks in the Neurobiobank. And there is no cost to the donor or the donor's next of kin. The bank makes all arrangements for tissue recovery. And that may even include paying the cost of transporting the body to the place where tissue can be recovered. In many cases, that is the actual funeral home where families want arrangements to be made. And so as we do not pay for funeral arrangements, we can pay for transport of the body when necessary. Another thing that makes our bank a little unique as a brain bank is that, yes, we do collect the brain, but we also, whenever possible, make an effort to collect systemic tissues as well. And so if we're talking about a disease where other organs or tissues are important in the symptomology, we will definitely make an effort to collect, certainly not the whole organs, but we will sample from as many organs as we can arrange the recovery technician to collect for us. And although pre-registration is best, it's always best to give us time to make arrangements to find somebody to perform the recovery, to get the consenting done well ahead of time. We can be contacted when death is imminent, or even if death has already occurred, but we're alerted in a timely fashion, right? That tissue has to be removed and it has to be preserved, ideally within 24 hours, but we do make exceptions when appropriate. But again, best to pre-register. But if pre-registration has not occurred, it's not a deal breaker. If death is imminent or has just occurred, it's not too late to contact our bank. Next slide, please. We do have inclusion-exclusion criteria. Again, we take donors of all ages. Infectious disease testing is not required, but we will do it. And if there are certain diseases present, we may or may not be able to distribute that tissue to researchers. We certainly would prefer not to have a history of exposure to things like HIV or hepatitis. And whereas a history of cancer is not an exclusion, if the cancer has metastasized to the brain, that is an exclusion criteria for us. A history of chemotherapy or radiation, I don't want to give it a hard no, but if it's happened recently, it might be exclusion. Sepsis is an exclusion criteria. Ventilator time is subjective. There's no really hard number here. The less time on the ventilator, the better for the condition of certain tissues, most importantly, the brain. Again, I mentioned the post-mortem interval after death. We shoot for 24-hour collection period, but for rare diseases such as SLO, we try to make exceptions when possible, and we'll extend that up to 36 hours if necessary. And of course, storage once the tissue is collected is very important. Next slide, please. So how to initiate registration? There are actually two options to do this. And of course, from our perspective, the easiest and most direct is to contact us directly. We have a website that's embedded within the University of Maryland's portal. If you just type in Maryland Brain and Tissue Bank, most likely you're going to get here. Once you get here, our page is divided into, we have a tab for researchers and a tab for families. You hit that families tab and you're going to end up on this page. You can hit that register button and you'll be taken to an online form where you can submit some preliminary information, and then you will be contacted and we will help you with the rest of the consenting process. Or you can call that number directly, 1-800-847-1539, 24 hours a day, 365 days a year. If it's after hours or it's the weekend, you will get an answering service, and that answering service will deliver that message to whichever one of our project coordinators is on call, and then you can expect to be contacted from that project coordinator, and we will determine how urgent things really are, if it has to be taken care of immediately or if we have a few days to deal with the registration process. Next slide, please. Tissue registration process, as I mentioned, you contact us either by telephone or through the computer. We then have to send you a consent packet, which includes the consenting document, the HIPAA form, which will allow us to make requests for medical records, and a questionnaire in which we ask you to complete as much of it as possible so that we know as much as possible about the donor's history. We can't really progress further than that until the consent documentation is returned to us. That can occur either through mail, fax, or as an attachment to email. And then we have to be alerted when the time comes. When the donor passes, we have to be alerted as soon as possible, and then we can put recovery procedures into action. Next slide, please. So I mentioned there are two options, right? And so if for some reason you don't remember that it's the University of Maryland Brain and Tissue Bank that you're interested in, but you do remember that the NIH has a neurobiobank, then by all means, go to this website. NIH Brain Bank will probably get you here. It's the NIH Neurobiobank. And this is the landing page. The Neurobiobank does not itself arrange for the donation of brains, but there's a foundation called the Brain Donor Project, which was founded specifically to aid in the registration of potential brain donors. You can see that blue button on the right-hand side. If you click that button, you're going to be put into contact with a volunteer at the Brain Donor Project. Next slide, please. What the Brain Donor Project does is it looks where you're calling from or where the recovery has to be made from, and it looks at the disease that we're talking about. And then it assigns that case to the appropriate bank. And even though the Maryland's catchment area is the Great Lakes and Mid-Atlantic region, for diseases such as SLO or rare diseases, they know to put that into contact with us. So wherever you are in the country, if you go through the Neurobiobank, chances are they're going to direct you right to the Maryland Brain and Tissue Bank. Next slide, please. Okay. The process, once collection has occurred, tissue is rushed, and we have tissue coordinators which preserve that tissue in a way that is optimal for all kinds of research, whether that's frozen samples or fixed. The brain gets examined extensively by neuropathologists, as it would if it was a clinical report. We do quality control on the tissue. All that data gets put into our central database. It gets reviewed by our own neurologists and physicians on site to help confirm the diagnosis. And then ultimately that data gets uploaded with identities excluded, redacted. That data gets uploaded to the Neurobiobank's portal, where researchers can search for tissue and find it and make the requests that are best for their research. And then of course, all the tissue we have is stored appropriately and made available to qualified researchers upon application. Next slide, please. This is actually the landing page. If you come to our website, you can see down at the lower left, that's the button for donors and families, the one on the right for researchers to give them more information. And I will just say thank you, and I greatly appreciate your inviting me to talk, and I wish you all the best. Thank you. Thank you.
There were no questions. Oh, there's some questions in the room though, Gretchen. Who was first? Here, Martha, you're closer.
Is it possible to do this in the UK?
I won't say it's impossible, but it's been some time since we've collected internationally. At the very least, we could probably try to put someone in contact with a more localized facility, and then a transfer might be able to be arranged where the tissue is sent to our bank through the aid of a material transfer agreement.
Thank you.
Two quick questions. One, I have guardianship of my children. So as far as the legal pieces of that, do you have somebody who walks you through, with the legal pieces, the guardianship letter and all of those things?
Yes, we have two what we call project coordinators, which handle all that type of thing. Our goal is to walk everybody through the registration process and make it as easy and as less stressful as possible.
Thanks. And then my second one is, so my son on his state ID, he's not a driver, but the state ID, I have him as an organ donor. Should I cancel that in this case because to divert the organs?
It's not necessary to cancel that. Brain donation is not included in organ donation because it's not a transplantable tissue. And this is not uncommon for us to collect tissue from donors that are donating other organs for either transplantation or research. But if it would be best to have as much advance warning as possible so that we can coordinate that procedure with the group that will be performing the tissue donation recovery. So there's no need to cancel that.
Thanks. A follow-up to that, and this is only because if I have a preference, I would, the brain donation to SLO would be more important to me than the donation of the body organs. Do you run into more trouble when you have to share nicely that way, or has it been pretty streamlined?
It is streamlined, but the brain does not take precedence because it's not being transplanted.
Yeah,
the groups that are doing the recoveries, and those are the ones we're asking to also remove the brain for us. It'll be moved down the line, and generally speaking, that doesn't put it outside of the PMI window. So it's workable.
Okay, thank you.
Thank you.
Do you suggest that we put all of this in our special needs trust?
I'm afraid I have to plead ignorance. I'm not familiar with that.
We were advised to have a trust in case we pass away. So in the case of whatever happens to our children, specifically our daughter with special needs, we have a special needs aspect to our trust. So in order to have legals,
Yes. So anybody who has the authority, the legal authority to make these decisions should be directed to contact us at the appropriate time.
The other question I have is that both, my daughter has autism as well as a Smith-Lemli-Opitz. My question is, if someone just has an autism diagnosis, do you want their brains too?
Absolutely. All of these disorders, behavioral, psychiatric, physiological, are of great interest to the bank, and autism spectrum disorder, those cases are extremely valuable to the research community. Yes.
Thank you, Dr. Blanchard.
Thank you.
I'm just a few minutes to tell you about how this came to be, that this conversation was started. It was actually started twice. We had never actually, it was something that, we knew about, But we actually had an inquiry at the foundation, and we didn't have a process in place. And through our medical advisory board and the connection to the University of Maryland's Brain and Tissue Bank, we were able to take an inquiry, 2 inquiries, amazingly, that happened within a short period of time. And we were able to create a process that, even though you can contact directly, there is a place on our website that talks a little bit about a donation such as this, and then an introduction to it, and a connection also that will go directly to the Maryland Brain and Tissue Bank as well. We received the phone call, the second phone call, from a family of Matthew Tice, and who is an amazing individual. I believe he was 45 years old and severely affected with SLO, but it had always been his mother and his sister, Allie. His life wish for making a donation of this type. And it was important to them for this to happen. And so we rallied the troops, and we were able to make the connection and make a successful donation, our first donation of tissue for research that they had never had before. But in addition to the grateful thanks that we have to Matthew's family and Matthew himself, because it was really Matthew and his desire, not his family. But also I want to acknowledge Cody, which was the first call. And when we rallied, we were within a few weeks of having the process in place. Cody passed away, but it was an extremely important step in the process of being able to accept the donation from Matthew. So we are eternally grateful to both of them that we now have this amazing process that can help research in the future for all of our kids. And I just want to ask for a round of grateful thanks to these families and the researchers for helping us do this. So thank you. And that's